All right, so welcome to week 3 of microbiology lecture. I will be covering chapters 6 and 7, which are dedicated to the basics of viruses and also nutritional requirements for microbial growth. So to get the ball rolling here, I will start off with an introduction to the microbe that we call a virus. And out of all of the ones that we will be learning about this semester, I think these are my favorite. They are the smallest out of the list of microbes along with prions or prions, and they're also the most elusive. So unlike eukaryotic cells, which has this one theory that a lot of scientists support for their origin, we're not really sure about when viruses arose in our history and how. So there's a lot that we still do not understand and that we're continuing to learn about them. And also we're continuing to discover new viruses as we speak. And the cool thing about a virus is that they're actually more ubiquitous than bacteria. So there's even more viruses in our ecosystem than bacteria. Now, like I said, there's really no accepted school of thought as far as the origin of a virus. There are a few theories out there, one of which claims that it arose before a prokaryote or a eukaryote, or after, or simultaneously, and there are a few lines of evidence supporting all of those theories, but not one that's well accepted by everyone, even though they are considered the most abundant microbes on Earth. And the one characteristic that's super important for all of you to remember about a virus is that it needs a host cell to thrive or to replicate. So in order to produce new copies of itself, it needs a host cell, which would be the animal or the human that is infected and whatever the target cell is. So influenza, for example, typical target would be the cells of the respiratory system, so it will replicate within those cells. Therefore, all viruses are what we call obligate intracellular parasites. Now, as far as the basic structural components of every virus, they have for sure a protein shell, which we refer to as a capsid, and also their own genome. So these are the two basic structural components, but they do not fulfill all of the characteristics of life. So they depend on a host cell. Some do not have a membrane. They're not able to carry out metabolism on their own or synthesize their own proteins. But just remember, they need a host cell. They have a protein shell, which we call the capsid, and they also have their own genome. Now, the genome, our genome, the human genome, is made up of DNA. For viruses, they could have either a DNA, deoxyribonucleic acid genome, like ours, or an RNA, ribonucleic acid. Now, what's the big difference here? It depends on as far as the way that the virus is replicating itself. There are extra steps that are involved here when you have a DNA virus, and I'm going to go through that in a little bit. Now, as far as the general size of viruses, they're much smaller than bacteria. So here, if you check out the scale, this is your basic E. coli, garden variety bacterium, about 2 microns long. Take a look at all of these viruses. So we're starting off with 1 going all the way up to 10, and here they're labeled. Pox virus is the largest known virus to cause disease in humans, and that runs about 250 nanometers. Once you start to go into the smaller structural viruses, you see here adeno-influenza is less than half of the size of pox, all the way down to causative agent of yellow fever, which is just a mere 22 nanometers, which is just a little bit bigger than an average hemoglobin. So we're talking pretty small scale here. And this whole cell that's housing all of these is a typical eukaryote, a yeast cell, which is around 7 microns. So remember, for viruses, that scope that was sent to you to your house along with your custom kit is a compound light microscope. You cannot use that for the online guys. You cannot use that monocular to see a virus. You need an electron microscope, which for either the online group or the hybrid that's with me in lab, we only have access to the light microscopes, not the electron. All right. So as far as other accessory structures that you would find within typical viruses. Now remember, what did I say that every virus must have? At least that protein coat and the nucleic acid or the genome, right? Also on the cover, you may find an envelope. So this is very similar to a cell membrane, phospholipid bilayer that you would find within eukaryotic cells and all prokaryotes. But we also may have within a virus some enzymes or additional proteins. Not all viruses have these, but it's worth noting here. And also it's worth noting that unlike both eukaryotes and prokaryotes that have ribosomes, viruses lack ribosomes. So because of this, this is why I refer to viruses as little molecular machines that hijack your cell's machinery, because they actually will use the host cell's ribosomes to make proteins. So let's talk about the basic structural elements. So the outer covering, this basic protein coat, is what we call a capsid. And the major function of this capsid is to protect the nucleic acid. If we're talking the nucleic acid together with the capsid, this is what we call the nucleo or nucleocapsid. Some viruses also you may find an envelope, and in this case it would be called enveloped. Without that extra layer, it would be non-enveloped or naked virus. And if you take this capsid, this protein layer, and you break it up into smaller units, they're all identical protein subunits referred to as capsomers. So the whole coat is a capsid, smaller units are capsomers. Now there's two basic structural capsid types. You can have a continual helical structure, which consists of basically capsomers that form this cylindrical-like structure, or you could have an icosahedral structure that has many sides. So in this particular example, it's showing you one that has 20 sides with 12 corners, but there can be variations upon this theme. So we either have, again, helical, which is more of a cylindrical structure of the capsid, or icosahedral, which has many sides. Now, if we have an envelope on the outer layer of that capsid, usually this is characteristically found in the majority of animal viruses, and this envelope will be acquired during the viral replication cycle when the newly formed mature virion leaves the host cell. So when it forms a new copy of itself, it's ready to exit that cell, it will actually pinch off part of the host cell's membrane or envelope. You also may find on the surface of the virus these protein components that we call spikes. All right, so if you see spikes on the surface of the virus, these are the structures that the virus will use to attach to whatever cell it is invading. And this is very important because, for example, there are some people who are immune to specific viral infections because they lack the receptors that allow for that unique interaction between the viral spike and the receptor. So if you don't express a particular receptor that's recognized by a viral spike, you may be potentially immune to a specific virus. So this actually triggers the initial step of viral replication. Now, as far as this envelope, if a virus contains this outer layer, it's an added layer of protection, and it also helps the virus bind to a specific host cell surface and assists, it enhances the viral replication cycle, in particular the ability of the viral DNA or RNA to enter the host cell. So this is a picture of a virus about to bind to a host cell via spikes on the surface. Right, and so we talked about how there are two general morphologies for viruses. Either you can have a helical virus or an icosahedral. There's also the potential for viruses to exist as what we call complex or atypical. And so the two that I would like you guys to be familiar with are pox viruses. Those are animal viruses that can infect both humans, and there's cowpox, monkeypox, even elephant pox. But the pox viruses are one of the largest out of all of the viruses that are known to man, and they lack a common typical garden variety capsid and are instead covered by a dense layer of lipo or lipoproteins. Another group of complex viruses are what we call bacteriophages or bacteriophages. And these are represented by this tripod-looking, spider-looking thing on the right-hand side. So this is a pox virus, a cartoon image here. This is an actual electron micrograph of a bacteriophage and a cartoon image of one of these as well. So they're complex in the sense of their structural elements. Again, here the pox has this dense layer of lipoproteins, and on the right-hand side here you see that the bacteriophage has these distinct portions of this entity. So you have the capsid head, you have a collar, you have the sheath portion, and you have these tail fibers. And so this thing is very similar to a lunar landing module. It literally will hop right onto the bacterial cell. It will attach via these tail fibers, and it will actually inject its nucleic acid right into the target bacterial cell. So unlike the smallpox viruses that infect animal cells, bacteriophage actually infect bacterial cells. So I know this takes a second to wrap your head around, but there are indeed viruses that are solely infective to bacteria. All right, so those are the complex viruses. Also, we talked about how there are enveloped viruses, and then there's non-enveloped viruses as well. So later in the semester, we'll actually go through and I'll cover every single viral family, and I'll talk about all of the infectious diseases that are typically associated with those viruses, the symptoms, the treatment, all of the characteristics of that particular virus. But just for now, we're just covering the basics. So just remember, there are some that are complex, atypical, some that have an envelope surrounding their capsid, and then some that are non-enveloped or naked viruses. Now, definitely every single virus will have a capsid, or it could deviate if it's a pox virus that has that lipoprotein layer, but it also will have nucleic acid. So every virus has a viral genome. Now that genome will consist of either DNA or RNA, but you'll never have a viral genome that has both. So later in the semester, I think it's the last 2 weeks, we're covering chapters 24 and 25, which are one is dedicated to DNA viruses and the other is solely RNA viruses. So they are a little different dependent upon how they will replicate their genetic material. But whether you are a DNA or an RNA virus, the information in this sequence, so DNA stands for deoxyribonucleic acid or ribonucleic acid. These are basically a stretch of letters. So the A's, T's, C's, and G's that make up our DNA, for example, are the same stretch of letters or sequence code that you find in a viral genome. And basically, these provide the information for all of the proteins to be synthesized and all of the RNA to be synthesized in order to make a completely new virus. So the DNA is actually the software, the code to write all the programs for how to make a completely new viral particle. It holds true for our cells as well. Our cells contain all of that information in our human genome as well. Now, the only difference here, if you guys take a look, so right-hand side here, you're seeing if you have DNA, the DNA then gets transcribed to RNA, and then the RNA is translated to protein. There's an added step here with a DNA virus. If you are an RNA virus, all you have to do is translate that information to protein, and you also have to replicate the RNA. But there are a couple extra steps here with DNA viruses, and they are different as well with regards to the location of usually where they replicate within cells. Now, as far as the number of genes that you would find in a DNA and RNA virus, it varies. So we'll kind of mention details about that later in the semester, but it can be anywhere from a few to as many as 100 or more. Animal cells, the human genome, for example, has 23 to 25,000 genes. So we don't ever approach that in the virus. They're fairly small. Now, if you are a DNA virus, like I said, remember, you have to transcribe this information from the DNA to make RNA, and then this RNA needs to be translated to make protein. DNA viruses, the majority of DNA viruses are what we call DS, meaning double-stranded. But there is an exception here where they may be single-stranded. So the single-stranded DNA viruses are the parvoviruses. For RNA viruses, they are usually single-stranded, but they may be double-stranded. The double-stranded RNA viruses are what we call reoviruses. But I would remember general trend, DNA is DS, RNA is SS, and sometimes we find DNA genomes that are circular. Also with RNA viruses, it may be segmented into what we call picoRNA, which is just basically small pieces of that virus. Also, you guys may see throughout your reading a DNA or an RNA virus, more appropriate to an RNA virus, that's labeled either positive or negative RNA, or it may say, analogous to that, sense or antisense RNA virus. Now, what that is referring to is that's referring to this strand of RNA that will be translated to make the protein. So if you are an antisense RNA virus, you need to convert to the complementary sense strand before that strand can be used as a template to make protein. If you already are the sense RNA virus, then you can skip this step. So just remember, sense goes right to protein, antisense, you need an added conversion step. Now, viruses, so we said that every virus will have at least a capsid, a protein coat, and also a viral genome, which is DNA or RNA. Additionally, you may find some preformed enzymes that could be present. So this is just as a reference here, this is a cartoon image of HIV. And you're seeing the parts that we just went over, right? So here's the genome. It's an RNA genome. There's two single strands of RNA. And you're seeing the spikes. This thing has an envelope around it, and it also has a capsid around it. Okay. Now also, HIV carries with it reverse transcriptase, which is down here, this little red guy, also integrase, and protease. So these are three enzymes that are preformed within this virus. Now, you may also have some viruses that bring along a polymerase, a DNA polymerase, which is basically an enzyme that will replicate DNA, or an RNA polymerase that will transcribe DNA into RNA, or you could have replicase, which will just copy RNA, and the reverse transcriptase that you see here, which is a special enzyme only found solely in retroviruses, will take RNA and convert it to DNA. So this is why HIV, once a person is infected with HIV, that information actually gets integrated into the host cell's chromosome, and that individual remains infected forever because it's actually in the genome. Now, I think that there's on the horizon a potential for that information to be snipped out of our cells via this technology called CRISPR, but that's down the line. So anyways, preformed enzymes, maybe polymerases, replicases, or RT, reverse transcriptase. Now, how do we classify viruses? So main thing to remember, guys, is that usually it's based on the genetic material. So even your textbook divides it up into chapter 24 and chapter 25, DNA versus RNA viruses. So that's really the first line of a characteristic that's used for classification. Also, structure. Does it have an envelope? Is it non-enveloped or naked? Does it have those lipoproteins, so chemical composition? And I would remember family name, it's always Viridae. So herpes viridae, for example, is a viral family. Within the herpes viridae viral family, there is a genus called simplex virus, which always ends in virus. Now, HSV1, which is herpes simplex virus 1, is within this genus. So this is a little different than how we classify genus, species for bacteria or any of the other microbes. I would remember the family, Viridae, and genus name is always virus. And this would be a subtype herpes simplex virus 1 or HSV1. Now, this is just to show you guys now all of the different families that we know of, and you can just kind of breeze through this table. I'm not going to require that you guys know this stuff for the first exam, but I would just kind of breeze through to familiarize yourselves with some of the terminology here, because we will be going back to this stuff later in the semester. So we'll be learning about all of these different viral families. Orthomyxoviridae, which consists of the influenza virus, swine influenza, rhabdo, so that contains rabies, coronavirus. So all of these we will be learning about the associated diseases, how to diagnose them, how to treat them towards the end of the semester. Now for the good stuff of how a virus actually makes new copies of itself. So when you contract a virus and you come down with a viral infection, you're sick, you're sneezing, you have a fever, you have the flu. What is the process by which that virus has entered your respiratory system through the nasal passages, and how has it infected your cells and replicated at such a high rate that now you have all these symptoms that are showing? Well, I'm going to go through the different steps here of the typical animal virus multiplication cycle. And I would like you to be familiar with these 6 basic steps. So the first one, and I'm going to show you a cartoon image in a second, so it's not as boring to just look at the text here. But the first step here is always attachment. So that virus has to enter somehow through a portal of entry in your body, and it has to bind to a specific target cell. So every virus, not only does it have a target host, which would be the type of animal, the type of organism that it is infecting, it also has a particular host cell type. So it will use its mode of attachment, which we went over, typically those spikes. It will attach to the target cell, and then it will uncoat and penetrate that cell by uncoating the capsid and releasing the nucleic acid. So the most important part of the virus is the nucleic acid to enter that host cell. Once the nucleic acid has entered the host cell, it now can hijack the host cell's machinery to make new viral components. Once those new viral components are constructed, it then will start assembling into mature viral particles, and it will release from the host cell via a process by budding, which is also called exocytosis, or through cell lysis. So the cell will actually pop and the virus will be released. And usually there are hundreds of new viral particles that are produced. So attach, penetration, uncoating, new components, they're assembling, they're released. So this is the cartoon image. So let's say we have an influenza virus here. So someone sneezes in your face that's infected with the flu and they don't cover their nose or their mouth, and you breathe it in. And those droplets contain the flu virus, orthomyxo, influenza type A. Now you breathe it in. That influenza virus uses its spikes on the surface to attach to receptors on your epithelial respiratory cells. And it will then penetrate through the cell membrane. So the virus is engulfed into a vesicle, and then it's going to uncoat. So it's going to release its genetic material. So here it's an RNA virus. So the nucleic acid is in the host cell. Remember, all this blue stuff here is the host cell, cytoplasm. So this is one of your respiratory epithelial cells. The RNA is in. Now remember, a virus is an obligate intracellular parasite. It has to uncoat its nucleic acid. It has to enter the cell in order to replicate and make new copies of itself. So now, in order to do that, how do we go from just this single strand of RNA to a whole new copy of this virus, or a whole new viral particle? Well, remember I said that the genome acts like software to write all of the programs, to write and synthesize everything that makes up this virus. So this RNA is going to be used as a template to make new RNA. A replicase is going to be used to make more copies of RNA. The virus is clever in that it's going to use the host cell's ribosomes, which can't differentiate between its own RNA or the viral RNA. It's going to translate this RNA into protein. So this would be the capsomer units, which make up the capsid, the spikes, maybe preformed enzymes. So all of the protein, all of the RNA, it's going to assemble into new viral particles. So it's not actually showing you it going together here until we have now here a strand of RNA and a newly formed capsid, and it's going to be released. So this is just one viral replication cycle of we're starting off with one virus, right? And we're making a new virion. But in the end, whenever you have one virus that enters, usually you have hundreds of these that are formed. And now as this buds off, it's going to take some of the cell membrane with it. All right, so enveloped viruses bud off, carrying away envelope with the spikes. So now this complete virus or virion is ready to now go and infect another one of your respiratory epithelial cells. So that is, in a nutshell, animal virus replication. Now a little bit about attachment uniquely for every virus. And I just mentioned this a couple slides ago, but every virus has a host range. So I said before, pox viruses, pox viridae, the ones that fall within that family can only infect certain hosts. So smallpox can infect humans, and there are deviations or variations of a smallpox virus like cowpox or monkeypox. But smallpox itself is only infective to humans. And also, along with the host range, it also has a particular host cell type. So for example, Hep B is only infective to liver. Polio is infective to intestinal and nerve cells. Rabies can infect various cell types. And it has to do with the spikes. How does it actually attach? So it's the spikes that are uniquely recognized by a particular receptor that is only found on certain cell types. Now, as far as how the virus may enter cells and uncoat its viral genome, it's good to know that it could be through this process that we call endocytosis, where the entire virus is engulfed and then enclosed in a vacuole or a vesicle. Or you could have fusion, where the envelope is merging directly with the membrane. And then this allows for its entry into the cytoplasm. So these are cartoon images of what I just went over here. So the variety, if you check out on the top here, you see that it's fusing with the cell membrane, and then the nucleocapsid is entering. The second one down, you see that the virus is binding, it's engulfed into a vesicle, and both the vesicle and the envelope and the capsid break down, and then you just have the free DNA. Or you could have a scenario where, similar to the one above, you just have the RNA that is released from the vesicle. All right, so those are all varieties of the process of how the nucleic acid is introduced into the host cell. Now, I would also like us all to remember, generally, DNA viruses have a preference to be replicated and assembled in the nucleus, whereas RNA viruses generally like to be assembled in the cytoplasm. So you might be wondering why this is the case. Well, remember, there's an added step here, right? I said, so for DNA viruses, you need to convert DNA to RNA first in order to synthesize protein. So you need the RNA polymerase, which is found in the nucleus, whereas you could just go right to the cytoplasm to start translating this RNA to protein. Now, for release, there are two ways: through budding or lysis. So in lysis, basically the cell dies, it ruptures, new viruses are released. Or in budding, also called exocytosis, the nucleocapsid binds to the membrane and then it pinches off from that membrane. The cell is not immediately destroyed. So just remember two varieties of viral release. Now, something worth noting here, and I'm going to come back to this at the end of the semester, is this whole notion of cytopathic effects with regards to viral infections. So even when we all go to the doctor, if there is a potential for a viral infection, a lot of times they will take a sample from the infected site and they will look at the cells under a compound light microscope. And they're not looking for the virus itself. Remember, I said viruses are very small. It's hard to see just using a light microscope. Rather, they're looking at damage to those target cells. So this is what they do, for example, in pap smears to look for HPV, human papilloma virus. They swab, they take a sample from the cervix, and they look at those cells under the scope to see, is there any dysplasia? Is there a change in the size or the shape of those cells? Are there any inclusion bodies? So these storage bodies within the cells, a high number of them. Sometimes viral infections can cause cells to fuse together. And when they fuse, you may have this one cell that has multiple nuclei in it. Or you may see cell damage reminiscent of cell lysis. So there's actually cell death. And this is the most severe case of a cytopathic effect here. Worst case scenario would be where you have a viral infection that actually can transform that healthy cell into a malignant one. And this is indeed what happens with some strains of HPV. There are these high-risk strains that target specific proteins within healthy cells that act as, you can think of some of the triggers analogous to a gas and a brake in the car. So cancer basically consists of a mass of cells, and especially malignant cancer contains a mass of cells that don't know how to stop growing. So in that cell, there are molecular triggers analogous to the gas and the brake in a car, whereby the gas is constantly on and the brake doesn't work. So there are proteins that act like the gas pedal, which are triggered by HPV, and the ones that are supposed to be stopping that growth or initiating the brake are also inhibited by HPV. And so you have these targets that occur or that are inhibited or activated, and in the end you end up with potential cancer out of the deal, which is again the worst case scenario. But this is a way to actually tell or provide some line of evidence that there is a viral infection rather than actually seeing the virus itself, which is difficult to tell when you're using a light microscope. And it's not the case where you have the same cytopathic changes for every viral infection. So smallpox, for example, you're going to see slightly different cytopathic effects than influenza, for example, measles, and like I just cited, HPV. They all have their own unique changes when you look under the scope. And there are a lot of viral infections that have the ability to display this persistency. So there are cells that are infected that can harbor the virus for a long period of time. They're not immediately lysed. Also, you can have chronic viral infections. So, for example, the measles virus can remain hidden in brain cells for many years. Herpes simplex virus, or the causative agent of, in this case, cold sores, HSV1, HSV2 is the causative agent of genital herpes. These can lie latent in specific nerves, the trigeminal nerve, and only to be later reactivated to stimulate what we call outbreaks of this infection, or even herpes zoster, same thing. The same virus that causes chickenpox also can be later reactivated to cause a different manifestation of the disease that we call shingles. So there's a lot of diversity along the spectrum here with regards to viral infections, and some do have this nasty potential to transform cells into cancerous ones. So cancer, if anyone's heard of the book, The Immortal Life of Henrietta Lacks. Henrietta Lacks was an African-American woman who died in the 50s of a particular, I want to say, uterine cancer or ovarian cancer, and her cells were harvested from her tumor after she died. So she died in the 50s, her cells were harvested, and the HeLa cell line is named after her, Henrietta Lacks, and it is the model for cancer. It is the model cancer cell line in all labs across the world. So you can Google HeLa, H-E-L-A cells, and these cells are still growing in a petri dish today. And it's just wild that these cells were found in this tumor, in this malignant tumor that this woman died from, and these cells have an increased rate of growth. They don't know how to stop growing. Their chromosomes, they almost have their own karyotype, so their own altered, mutated genetic identity, and they have the capacity to divide for indefinite time periods. And they're being used again to study all different types of cancer treatments and just the whole model for cancer itself. Now, what's scary is that there are a lot of viruses that we're now discovering have the potential to predispose healthy cells to cancerous ones. So I cited one already, HPV, which is papillomavirus. There are certain HPV strains that are known to predispose an individual to cervical cancer. Also, hepatitis, for example, Hep B, Hep C can predispose an individual to some forms of liver cancer, hardening of the liver. Also, EBV, Epstein-Barr virus, which is surprisingly a ubiquitous virus. This is one that by the time we are 50 years old, 95% of the population is infected with this, but in certain circles, it is known to induce this form of cancer called Burkitt's lymphoma. And this is usually in young African-American children who are also co-infected with malaria, but we do see the potential for it to exist as an oncovirus. So if it has anything to do with cancer, usually you see this onco in front of it. So oncovirus, cancer causing. Now, I'm going to switch gears here from animal viruses into the viruses that infect bacteria. So remember, bacteriophages or bacteriophages are unique to bacterial cells, not animal cells. So these guys can infect our human eukaryotic cells. There are bacteriophages that could infect some of the bacteria that live in our gut, right? I talked about the commensal microbiome. So there are some phages that could infect those cells. The most widely studied are those that infect E. coli. And what I would like you to remember is that bacteriophages are specific to certain types of bacteria. So not all bacteriophages can infect every bacterial cell. So they are unique in that sense. And what's really cool about a bacteriophage is structurally it's complex. Remember, it looks like that lunar landing module with those legs and the capsid head on the top and that sheath that actually acts like a syringe. So I'm going to go through the steps of the cycle, which I hope you guys will pick up on. That's very similar to the animal viral replication cycle. There's one added step here that I'm going to cover, but kind of the same process, right? So we have attachment, penetration, replication, assembly, synthesis or maturation, and then release, right? So almost the same process here. The only thing that I'm going to add to the bacteriophage replication cycle is the lysogenic state or the lysogenic cycle. So when a bacteriophage, starting at step 1 here, here's this little lunar landing module bacteriophage, it hops on to the bacterial cell. Okay, this is a single bacterial cell. This is the virus. And remember, all it really needs to do is just inject the nucleic acid. Now, the nucleic acid is present in the head portion. And what is the circle here? This is the bacterial cell's chromosome. So bacterial cells have their own DNA, and it's usually circular in the form of what we call a chromosome here. So first step, the bacteriophage hops on, it attaches, okay? Oh, we're going this way. It attaches, and it's going to inject its nucleic acid. So you see the viral DNA is entering the cell, all right? Once it enters the cell, you have duplication of these components. So you have replication of this DNA, you have the DNA that is then translated to RNA, the RNA is translated to protein, so it's DNA transcribed to RNA, RNA is translated to make protein, and we see all of the components here that make up the bacteriophage, right? The capsid, the tail fibers, the sheath. These are all synthesized from the information, the sequence that's in this DNA here, all right? So now we make new bacteriophage. We have mature bacteriophage. The cell lyses and the bacteriophage are released. So this is what we call the lytic cycle. Whoops. This is very similar to what I just showed you guys before with the animal viral replication cycle. But there is an added step here. So if you guys take a look on the left-hand side here of the slide, if you look at the step following the introduction of the nucleic acid into the bacterial cell, something different is going to happen here. So this blue piece of viral DNA is now going to be integrated into the bacterial cell's chromosome. And once that becomes integrated, it becomes latent as what we call a prophage. This is the lysogenic state. Now, usually the viral DNA is inserted at specific sites along the chromosome. And what happens here is now when this bacterial cell goes to divide and replicate its DNA, it is going to replicate the phage DNA as well. So even though you don't have new bacteriophage produced, you still have all of the information to make new bacteriophage in the chromosome. And what I will say is this lysogenic cycle has allowed some bacteria to become more pathogenic or more highly disease-causing than others. So diphtheria, for example, has a toxin that is produced, the bacteria that causes diphtheria has a toxin in its bacterial chromosome that was introduced by a bacteriophage. So it can actually enhance virulence or enhance pathogenesis. So very similar steps here with regards to viral replication or viral multiplication when we're looking at how a bacteriophage behaves versus an animal virus. So same steps. The only difference here that I can really point out is that, remember, with the bacteriophage, you just have the nucleic acid that enters. The rest of this thing does not enter the bacterial cell. All right, so it's just the viral nucleic acid, whereas I showed you guys with the animal virus, you may actually have the whole thing that's engulfed in a vesicle. Also, the synthesis and assembly for animal viruses could occur in the nucleus. Bacterial cells don't have a nucleus. And there's this viral persistence that can occur with the bacteriophage known as the lysogenic state. All right, so pretty similar, but good to know the minor differences. Now remember, lysogenic conversion, I cited an example, diphtheria, where you have the phage genes, the bacteriophage genetic information that can cause the production of toxins or enzymes that can actually cause bacteria to make people sick. So diphtheria, even cholera, which is a waterborne illness, or Clostridium botulinum. This is the bacterium that causes botulism. All of these have been enhanced as far as their virulence by this process. All right, so going back here, just remember lytic, we're going to attach to the bacterial cell. We're just going to introduce the genetic material. We're going to make new copies, and the cell's going to lyse. These guys are going to be released to go and infect new bacterial cells. Or we can just have attachment, release, right, into the bacterial cell and integration into the host cell's chromosome, which would be the lysogenic state. How do we grow viruses in the lab? This is very different than growing, let's say, a bacterium or even algae or protozoa in the lab. In order to grow a virus, remember, it is an obligate intracellular parasite. You need a host cell. So I would like you guys to remember that you either need a cell line, okay? You can actually, in vitro, in a petri dish, infect cells and look for cytopathic effects, or you could use embryos, bird embryos, or sometimes they will use live animals. So actually infect or inoculate a live animal and allow the virus to replicate via that method. So here we have the embryo, cell lines, looking at cytopathic effects. You also can infect bacteria with bacteriophage, a lawn of bacteria on the surface of the plate, and look for clearing on that plate. So wherever the bacteriophage have infected those bacteria, you would see these patches or these zones as a result of lysis from that infection. Now, just some fun facts here about viral infections. So viral infections are generally more difficult to treat. A lot of them can cause serious side effects, even though they are the most common cause of acute infections. There's actually several billion viral infections per year. And some viruses, unfortunately, are associated with these high mortality rates. There are a lot of viruses that are on the list of emerging infectious diseases. So these are diseases that are on the rise. We don't have an appropriate treatment or cure for. There are also more that we're adding to the list nowadays. connecting viruses to chronic afflictions of unknown cause, so like chronic fatigue syndrome, for example, or even some forms of cancer. We're finding an infectious link, a lot of viruses linked there. And they do indeed play a major role in the Earth's ecosystem. They are the most ubiquitous, even over bacteria. And I wanted to, since we're on the topic of viruses, I wanted to at least mention this more closely related microbial partner, more closely related to a virus than to any other microbe that we will talk about, and that would be the prion or the prion. Now, I say closely related because of the fact that they are just as elusive, if not more elusive. I would say even more anomalous than a virus, and they're even smaller. A prion is literally just a protein. So just like a virus, it's acellular, but as far as the material that makes up a prion, it's literally just a misfolded protein. There's no nucleic acid, and these entities are extremely resistant to usual sterilization techniques, and they cause these awful neurodegenerative diseases. One example here, if you guys have heard of mad cow's disease, there is a human form of this. And this is what we call a spongiform encephalopathy. It's called spongiform because when you look at the brains of animals or humans after they've been infected with an infectious prion, the brain looks like a sponge because it literally eats away at the brain. And this is encephalopathy for the target. All diseases associated with infectious proteins or prions are known to be fatal. There are no treatments, no cures. And this is actually an electron micrograph of a prion fibril. Kind of similar to Alzheimer's disease as far as the pathology. So in Alzheimer's disease, you have a buildup of plaque of these fibril like proteins, amyloid beta in the brain. This is similar. It's a misfolded protein, and it seeds all of the regular folded prion protein into the misfolded form, and it causes these fibers to build up. So prion diseases, the first one actually was found in sheep, and it was called scrapie because the sheep would scrape all of their hair off of their body as the prion was eating away at their brain. Also, there's the mad cow form, which is the bovine form. Also, wasting disease in elk, and even a human form, Creutzfeldt-Jakob's disease, which displays similar symptomatology as all of these others. In the human form, when people are diagnosed, usually they have 3 months to live. It's awfully neurodegenerative. You lose your ability to speak, to move, to see. You, similar to Alzheimer's, you forget everything, and you have, again, 3 months to live, and there's absolutely no cure, 100% mortality rate. So these are prions. Additionally, just to mention here, satellite viruses and viroids. So satellite viruses are very interesting. They actually depend on other viruses in order to replicate. So there's an adeno-associated virus. It only replicates in cells that are also infected with adenovirus, or the delta agent is another example of this. And also a viroid. So a viroid is unique in the sense that it's just a short stretch of RNA. So it's kind of similar to a virus in that it has one of the components, the nucleic acid, but there's no capsid, no protein coat. There's an electron micrograph of a few of them here on the left. This is one found in potatoes. So these have only been identified in plants. So these are some other non-cellular infectious agents. And I'm going to switch gears here and now talk about Chapter 7, which is dedicated to microbial nutrition. So microbes are extremely diverse with regards to the types of chemical substances that they can use to power all of their required processes. Now, I'm going to go over some terms here. So I know this slide is text heavy, and then we're going to get into how to actually grow microbes in the lab. And for the online crew, you guys have already been doing this at home. So you've seen the types of materials that you're working with, the agar, and also you've seen how long it actually takes for you to visualize the growth. So we're going to talk about all of the stages of the generation time for bacteria and etc. So basics with regards to terminology for nutrition. So there's two categories here of essential nutrients that all cells require for growth. That would be the macro and the micro. Macro, they're required in macro amounts, so large quantities, and they play critical, integral roles in cell structure and metabolic processes. So these would be the bulk proteins, the bulk carbs. Smaller quantities are required as well of the micronutrients or the trace elements. These would be involved in maintaining the structure of DNA, for example, the structure of specific enzymes or enzyme function. So this would be manganese, zinc, nickel would be a micronutrient. Also, out of both of the essential nutrients, you could have organic or inorganic forms. I would just remember organic always contains carbon and hydrogen, so those carbs, lipids, proteins, versus inorganic, which basically contains collective aggregation of different atoms or molecules than carbon and hydrogen. So metals, salts, gases, also water. And also we have growth factors that are required in the form of as a nutrient. This would be because of the fact that the cells don't have the ability to synthesize them on their own. So it's just like our cells. We need to actually extract and absorb specific growth factors from the foods that we eat to drive our cellular processes. So these essential amino acids are critical. There's 20 of them, and I'm not going to require that you know all of them, but the amino acids make up each protein linear structure of every protein that exists. Also vitamins as well. Now, if you were to look in any cell consists, the majority of it is made up of 70% water. So water is the universal solvent. You're also going to find proteins, and the majority of that cell contains 6 elements. I would remember these 6: carbon, hydrogen, oxygen, phosphorus, sulfur, and nitrogen. We're going to be coming back to how some of these elements are fixed by microbes in the biogeochemical cycles. So there are free forms, gas forms of carbon, nitrogen, sulfur that are converted to fixed forms by microbial processes, and these are critical for recycling these elements in our environment. Now, I mentioned that microbes are extremely diverse with how they can carry out their basic metabolism. So there are some microbes that function as either a heterotroph or an autotroph. And as far as their energy source, they can even use light. They can gain energy through photosynthesis. Or a chemotroph gains energy from chemical compounds. So for the actual carbon source, an autotroph is an organism that uses carbon dioxide, and it's not nutritionally dependent on living things, so this would be the primary producers. Heterotroph would require carbon in an organic form, right? So these are just differences with regards to how some microbes use carbon, what the actual source of it is, and the actual source of energy, whether it's through chemical compounds or through light. Now, for the autotrophs using light, there are some that require oxygen or without oxygen. There are actually some that live in rock that survive totally on inorganic substances. Methanogens can even produce methane gas. If you look at the spectrum of diversity, I mean, it really is fascinating. There are microbes that can grow in the places that you would least expect any form of life, miles down in the ocean, in an environment where there are freezing temperatures or extremes of heat, extremes of pressure, or these other inorganic substances that we obviously cannot use to carry out our metabolism. Now, for heterotrophs, there are two categories. So saprobes are free living, and they typically will feed on organic material from dead organisms. And there's also parasites. So parasites will always derive the nutrients from the host. So majority of, we'll be talking about kind of three groups of parasites, protozoa, helminths, and ectoparasites that cause disease. And some even may be considered obligate parasites. They need the host to derive those nutrients for their energy. Now, since we're on the topic of nutrition, I brought up this idea that cells are 70% water. It's really important to talk about how materials, specifically water, are transported across the cell membrane. So osmosis is a huge concept here that you'll be asked, questioned about in the lab portion, also on the exam, and it's important to know the basic definition here, that osmosis is referring to the movement of water across a semi-permeable membrane from an area of high to an area of low concentration. So this is freely moving, so water always will move down this gradient. Facilitated diffusion will require a carrier, so this is different than the movement of water. And even more differential here is active transport, which requires energy. So this is always requiring ATP. So it's not just freely moving from an area of high to low. So basics of osmosis here. So I just said an area of high concentration to an area of low concentration, how molecules will diffuse, in aqueous environments. Now, this is the standard diagram that you always see here to describe the principle of osmosis. So you have this membrane sac that's filled with a solution that has solute, high amount of solute present in it. And what you see here is that there's more water molecules outside of this cellophane sac than there are inside. And so what's going to happen here is it's going to go from an area of high concentration to an area of low. So if you take a look here, water is flowing from the outside of this bag to the inside, and you see the level goes up here. Now, there are three possible dynamic scenarios with regards to changes in osmotic pressure. So if you have a cell that has an equal amount of water outside of the cell and inside of the cell, the rates of diffusion are equal. So you don't have an increase in or out either way. You may have the potential for a hypotonic environment. Now, in this case, the way I remember it is hypo for hippo, that the cell will actually swell and become larger. And this is happening because there's more water on the outside of the cell than the inside. So usually, it's very similar to what I showed you here. There's a high amount of solute inside, high amount of water outside. And then on the contrary, in a hypertonic environment, and this is what is exploited for example in preservation of foods, jams and jellies, or treated meats with a lot of salt. They have a high amount of solute, and this causes the water to leave any potential contaminant cells, and the cell will dry up. So it's a good food preservative. The cell's drying up because the water is leaving the cell. There's a higher concentration of water inside than outside the cell. And I would like you guys to remember the names for these processes of drying up. So the cell has become desiccated. This is referred to as plasmolysis or plasmolysis. If the cell swells and pops because of this hypotonic environment, this is referred to as plasmoptosis. So plasmoptosis, plasmolysis in the hypertonic scenario. Now also some other terms to bring up here for cells that are bringing substances into their internal contents through a vesicle. If they're ingesting other substances or cells, this would be phagocytosis. Penocytosis would be in the case of ingesting liquids. So you see penocytosis on the right and phagocytosis on the left. So phagocytosis is carried out by even our white blood cells. So our macrophages will attempt to, in response to a viral or a bacterial infection, they will actually engulf the pathogen into a vacuole and try and degrade it as an effort to destroy the infection. Peno, again, is liquids. Now I'd like to go over all of the different environmental factors that can influence how a microbe can grow. So again, for the online crew, you've been kind of playing around with inoculating plates and placing them as recommended at a higher, hotter temperature. And also for some of them, the ones that require more or less oxygen, maybe a little prickly with how they grow because some are very particular with the oxygen requirements. Some also like certain pH levels. They like more acidic environment or more basic environment. Also, osmotic pressure we just talked about, and barometric pressure is another factor. So with regards to temperature, every microbe has this scale of an optimal temperature with which it can grow, a max temperature, and a min temperature. So below the min and above the max, the microbe will grow. And right in the middle is the optimum temperature. So if you guys take a look here of the plotted psychrophile, the plotted psychrophile has an optimum temperature of about 5 degrees Celsius, right? And the max would be at about 15, the min would be minus 20. So rate of growth is on the Y, temperature on the X. Take a look at the mesophile. Optimum is around between 35 and 40 degrees Celsius. Min is 10, max is 50, and so on. So thermo likes to grow at very high temperatures. Psychro grows at below 0 degrees Celsius. Now, microbes are picky as far as the amount of oxygen that they prefer. So these are definitely terms you guys should remember. And it gets a little tricky with, I would say, facultative anaerobe, the aerotolerant, and the microaerophile. So make sure you remember the differences here. So an aerobe, think of air, it utilizes oxygen. Obligate aerobe, it can't grow without it. Right? So if there's no oxygen, you will not see any replication occurring. On the contrary, an obligate anaerobe cannot survive in oxygen, right? So these guys are very particular, okay? No oxygen, needs oxygen. Or you could have one that could go either way. Facultative anaerobe utilizes oxygen, but also can grow in its absence. Or the aerotolerant, which doesn't utilize oxygen, so it's more anaerobic, but it can survive and grow in its presence. So these guys are kind of opposite. And then the microaerophile, only a little bit. So it requires a small amount. And the anaerobe, this is just the basic, does not utilize oxygen. So if we were actually talking about all of these different terms here in inoculating, culture tubes, if you're looking for and measuring different amounts of oxygen that were required, you could do this in special liquid culture tubes, but also in solid culture, and you could look for microbes growing at the bottom, lacking oxygen, or throughout the whole span of the tube, so facultative, or they need oxygen, aerobic, right, obligate, aerobe here, or microaerophile would be right in the middle, so it needs a little bit of oxygen. And this is just an anaerobic chamber here where there's no oxygen. There are some that require higher amounts of CO2, so this would be referred to as a capnophile. Also, there are some that like very acidic environments, like our stomach, grow at low pH or high pH. And the majority of them like to grow between a pH of 6 and 8, so a neutrophile. Now, extremes of osmotic pressure, halophiles like a high concentration of salt. So remember, this would be a severely hypertonic environment, or some are osmotolerant. They do not require a high concentration of solute, but they can tolerate it, versus a barophile, different with regards to osmotic pressure, but these guys under high ATM or high atmospheric pressure levels, like 3 miles down, right, in the ocean, can survive under extreme pressure and actually will rupture if you take them out of that particular environment. Now, since we're on the topic of nutritional requirements, it's good to point out this notion of microbial association, specifically symbiotic ones. So there are 3 types of symbiotic association. Symbiosis or symbiotic association is just a fancy word or way to describe 2 organisms that are living in close association with one another. And they are required by one or by both. So in mutualism, both are dependent, both benefit. Commensalism, the commensal benefits, the other member is not harmed. And then parasitism, the parasite is dependent and benefits, whereas the host is harmed. There's also these associations, but the organisms are free living and they're not required for survival. So this is different than the symbiotic. Non-symbiotic, they're synergistic or antagonistic. So in a synergistic association, members cooperate and they're sharing nutrients, and then antagonistic, there's a negative effect, so they're inhibited or destroyed. So hopefully you guys can think of a couple mutualistic or commensalistic associations within the human body, right? So I stress this whole concept of the normal microbiome, right? So there are these hundreds of thousands, millions, billions of bacteria in our gut that display this mutualistic benefit, this mutualistic symbiotic association, where we need those bugs for vitamins, for our digestion, for our immune system, and they need our guts for the space and the nutrition that they get, and also the other bugs that are living there. And even for bacteria, you take them out of that environment. A lot of times you see, for example, in the gut, the majority of bacteria that actually are growing in your gut, you could not remove from the gut and grow on an agar plate, because of the fact that there are so many complex, it's such a highly intricate, complex ecosystem in your gut that we still don't even really understand all of the symbiotic associations in that environment, but we know that they are in high number. Now, whenever there's a microbe that actually makes you sick, we refer to this as a parasitic symbiotic relationship. And also I mentioned these synergistic or antagonistic. So this would be an example of a synergistic, non-symbiotic association between Azotobacter and Cellulomonas. So you see the ammonium is used by Cellulomonas to produce glucose, which then is used by Azotobacter to produce the ammonium. So it's a synergistic association. Now also here, since we're on the topic of nutrition, it's good to be familiar with not only the normal microbial flora, which I've stressed to you guys, and I love this image here that says, we are inhabited by as many as 10,000 bacterial species. These cells outnumber those which we consider our own by 10 to 1 and weigh about 3 pounds, the same as our brain. Together they are referred to as our microbiome, and they play such a crucial role in our lives that scientists have begun to reconsider what it means to be human. So, again, there are so many details of these associations that we're still learning about. So I think it's just such an exciting time for you guys to be studying in this field. But biofilms. So kind of connected to this concept of the normal flora that resides everywhere, right? Basically all throughout the span of our colon, all over the surface of our skin, nose, mouth, throat. We're even finding in the bladder, which we thought that was completely sterile, the lungs as well. What can happen is sometimes you can get the formation of this highly complex population of microbial, usually bacterial cells, that we call a biofilm. So basically a biofilm contains the population of bacteria that have the ability to break off from this complex layer and form new biofilms in that local environment. And the point with the biofilm that's really, really important to make a note of here is that this population of cells, once it actually starts to grow, it behaves like its own animal. So when these cells land on a surface here, whether it's your teeth or a catheter or a medical device or even a valve, a heart valve, or the inner ear or bone, what happens here is they're behaving when they're by themselves as a single organism, right? Single bacterial cell. They start plopping onto this surface, and when they start to grow, they start behaving like a multicellular organism rather than a single cell, and they become highly resistant to antibiotics, highly resistant to any antimicrobials. They actually dominate the structure of most natural environments on Earth. And I just pulled up a list here of a bunch of different infectious scenarios that have implications for the formation of biofilms. So here's a picture on the surface of a catheter, this biofilm. Also, here's a picture of the inner ear. So an ear infection can be caused by the buildup of a biofilm. Gum disease in chronic wounds, stubborn wounds that won't heal. Even cystic fibrosis, a buildup of a biofilm in the lungs. An atherosclerotic condition. Medical devices and implants are the worst for this. Catheters especially, even in the bone, UTIs in the bladder, in the bladder wall, that's a big problem. Also even kidney stones. So biofilms, again, they're highly resistant. Now, with regards to how microbes grow, I'm going to zoom in on and focus on bacteria. So bacteria divide by this process called binary fission. So what happens in binary fission is the parent cell will enlarge, it'll duplicate its chromosome, and it'll form what you see in this picture here, a central transverse septum dividing the cell into two daughter cells. So this is basically, that picture I showed you was this stage of it, right? So we're almost ready to divide into two new cells. But before that, this process of binary fission, this is bacteria. The chromosome will replicate it enlarges and separates into a whole new cell, and then you have the cells, the daughter cells that will divide. Now, how do we measure how bacteria grow? So specifically, the time that is measured, time required for a complete fission cycle here, so just this one cycle, is called the generation or doubling time. Each new fission cycle increases the population by a factor of 2. And for bacteria, generation times vary from minutes to days. And so you can actually, as you guys seen, you've inoculated your agar plates, and I bet you've seen a little bit of something growing on the plate after about a day, but you don't see the fully formed colonies for maybe 2 days because of the fact that it's not, I'm sure you're placing them somewhere in your house. If you were to place it into a very warm environment, they would grow quicker. So usually bacteria like around 37 degrees Celsius, which is higher than room temperature, which is around 60 to 65. How would we calculate population size over time? So I'm not going to require that you guys do this, but just to give you a heads up on this, how it's calculated, this is the total number of cells here, and it equals the starting number times 2n, and n denotes the generation time. All right, so that would be population size over time. Now you can actually count and calculate the generation time by doing a viable plate count. All right, so what you would do here is you could take an inoculated flask. So you have a flask of sterile media, you inoculate it with a few bacterial cells, and then you track it over time. So we're starting at 60 minutes all the way up to 600 minutes. So you see we start off with clear media that's not turbid, there's nothing growing in it, and progressively it gets cloudier and cloudier. And what you can do is you can plate at each stage a small volume and count the number of colonies that you see. So the plates are incubated after you inoculate each plate with around 100 microliters. And you record the number of colonies, CFU for colony forming unit, and then you can calculate the cell population according to the number of colonies that you see. Now, for sure, please remember this diagram. So this is the diagram for bacterial cell growth on a log scale over time. There are 4 basic stages here. There's the lag stage, the log stage or exponential, the stationary, and then the death phase. So this graph is representative of at time 0, and this is on a log scale. At time 0, we inoculate, let's say, a flask or a culture tube that has sterile media in it. And then over time, we plot the number of cells. And as you see, it slowly starts to increase, and then we get an exponential log phase growth. It starts to flatline here, and then it starts to go down. So what's happening at each stage? Well, in the lag phase, the cells are getting ready for cell division. They're synthesizing all of the macromolecules that are needed for binary fission, DNA duplication. They also may be synthesizing secondary, pre-secondary metabolites to make room for more space in their environment. So this would be antibiotics, for example. And then they enter the exponential growth phase, which is a log phase of growth. So they're dividing at an exponential rate. And then they reach the stationary phase here, where the rate of death equals the rate of the cells growing. So you don't have an increase in growth here. Half are dying, half are still growing. There's not any space left here once you get to the end of the stationary phase. So they've used up all the space. It's a very cloudy, turbid culture. There are no nutrients left. The cells start to die. So there's the rate of death here is superior to the rate of growth. And they will start to die exponentially. Now we can measure cell growth over time by measuring how cloudy a tube is. So you can kind of plot, you can do this plating method, or you can actually measure over time how cloudy does the tube get. So this cloudiness, another word for this scientifically, is how turbid the culture is. And we use this instrument called a spectrophotometer. It actually will shine light directly on the culture. And if it's clear, which we see here, so it's not cloudy, this looks like sterile media. There's nothing growing in it. And the spectrophotometer will actually tell us the percent of light that is transmitted. So how much light is going through. The higher amount of light that's transmitted, the less bacterial growth that you have. Once these transmission levels go down, right, so we see this level going down, that's because the light is being absorbed. So it would be more absorbed here, less absorbed. More transmittance on the left-hand side, less transmittance on the right-hand side. And that will give us a quantitative measurement of how many cells are in this culture. We also can actually take a sample out of the culture tube. So take a little bit of this, a small microliter sample, and place it on a little cell counter slide, and we can actually look under the microscope and count the cells. You could also do a colony count, which I showed you a few slides before. So these are more, in this case, this is more of a qualitative measurement. These are actually more quantitative because you're actually getting specific numbers for the amount of cells that are in that sample. So this is the last slide that I have for Chapter 7. I just wanted to make a note that for the online crew, please be keeping up with the experiments. There are a few that you need to actually start the weeks before the assignments are due, so please keep a lookout for the schedule on the syllabus, and you guys are doing a great job with your discussion posts. Just please make sure for the hybrid and the online that you provide citations and try and stick to the sources that I recommend, because some of these random sources I can't promise you that it's accurate information. And if you guys hear about anything on the news or anything that's really interesting to you that's in current day, an outbreak of an infection or a new treatment or a new discovery, share it with the group so we're all on board. And I hope everyone's doing well up until now. Please send me any questions via email or on Blackboard, and I'll see you guys next week.